Okay, well, firstly, I'd like to say thank you to the organisers for this excellent meeting, and I'm very glad to be here taking part in it. My name is Philip Rodley, and I'm the CEO of Neko Pharma. And before I founded Neko Pharma, I spent 13 years at Daiichi Sankyo, where I worked in various projects developing technology platforms in antibody and novel protein engineering. So Neko Pharma is a bioventure which is developing a novel platform technology to solve the biggest problems of drug efficacy and toxicity in multiple modalities. Common barriers to drug development include drug resistance, various toxicities related to poor specificity, difficult to reach and undruggable targets, and engineering problems. So the reason why these problems exist is because they really haven't been solved yet. So all of these modalities have various drawbacks and disadvantages which result in unacceptable toxicities or lack of efficacy, or they fail to address important things like disease relapse. And a failure in any one of these areas can result in a failure of drug development and a loss of revenue. So for example, if you look at SCFVs, everyone who's dealt with these knows that they have serious problems with expression and aggregation. And many of these SCFVs, if they're engineered into bispecific molecules, for example, the early stage candidates will simply drop out because they cannot be optimized. So this is obviously not an ideal platform. And also other single domain platforms such as protein scaffolds like anticalins and nanobodies, these are reliant on disulfide bonds for their stability. And as a result of that, they can't really be used for things like drug conjugation, and they cannot be used to target intracellular targets. So they're not actually very effective if you're trying to treat cancer. And another problem with these scaffolds is they have a restricted target binding. So for example, DARPins prefer to bind to convex surfaces because they're very rigid molecules. And in the case of anticalins, the randomization surface is around the edge of a cup-shaped molecule. So the target actually has to fit inside the cup in order for the anticalin to bind properly. So this leads to some quite serious disadvantages, for example, lack of functional activity or inability to target difficult targets like GPCRs. And this binding surface problem cannot be fixed. So the group which developed the DARPin scaffold, they actually tried to fix it by introducing a loop in the center of the DARPin binding surface to enable it to bind to a variety of drug targets. But the resulting instability and aggregation meant that this was never commercialized. So Neko Pharma's solution to these problems is a novel technology which we call Kazan body. This is derived from a volcano. It's stable, disulfide-free, it's easy to produce and engineer. For example, all of the binding clones that we isolated from our library have a thermostability above 80 degrees. And in addition to that, we have a very flexible binding surface of 3 randomized 10 amino acid solvent-exposed loops. And I think this is actually a world first, where it's possible to combine this level of binding surface flexibility with this level of stability. This can be easily engineered to bispecific format, and it can be efficiently produced in E. coli. The scaffold has been deimmunized, it's highly monomeric, disulfide-free, high affinity binders can be isolated directly from the library, and it has a favorable PK. So there are multiple applications arising from these Kazan body properties. For example, the easy multispecific engineering enables applications exploiting this multivalency, such as blood-brain barrier transcytosis, targeting of multiple spike protein variants on coronavirus, and overcome the antigen escape-mediated relapse of CAR-T. In addition, the disulfide-free status enables intracellular targeting and precise drug conjugation. So for example, using the Kazan body in a bioprotec format enables you to extend the druggable intracellular targets beyond those which are targetable by small compound drugs. This reduces things such as off-target toxicity, which is commonly observed in ordinary PROTAC molecules. This is an example of a bispecific drug conjugate. So we were able to rapidly construct highly stable and site-specific drug conjugates. These could be efficiently produced in E. coli, and they had a similar in vitro efficacy to a recently approved blockbuster ADC. So this is our business model, and as a platform technology provider, this allows us to develop multiple drug candidates against with multiple modes of action in multiple therapeutic areas to give us multiple shots on goal. This is our collaboration model. In this model, a drug company gets exclusive drug target nomination agreement, ownership of all of the cousin body drug leads against that target, and freedom to develop them into any modality that they like. And in exchange, Neko Pharma gets target nomination fees, milestones, and royalty payments. So this is just a snapshot of how much global pharma spent on early stage drug discovery last year. They spent about $10 billion on biologic lead discovery. And this is the market that our platform technology is targeting. And as you know, biologics make up about half of all drug sales. The top 5 biologics sold for $64 billion in 2021. So there's clearly a lot of money to be made. So our target market is the global top 20 drug companies which are developing next generation modalities. And we have a hybrid business model whereby our platform technology revenue will be used to fund our internal drug development pipeline. We believe we can develop multiple drugs against multiple disease targets using this versatile technology. So this is our management team. So our management team is made up of people who have experience in R&D, business development, medicine, and law. So myself and Dr. Kawashima-san, we both have extensive experience in pharma, in research and business development. And our medical and legal advisor, Robert Neller, he has extensive experience in law and in medicine. Thank you.
Thank you very much, Mr. Roderi.